So imagine you have a massive house fire.
Okay, terrible scenario, but I'm with you.
Right. So the emergency crew rushes in and they manage to put out the flames. And to fix it, they patch up these gaping, charred holes in the walls with like quick, dry cement.
Which works in a pinch.
Exactly. In the short term, it does the job. It holds the house together. But they use way too much of the cement. And now your house is incredibly rigid. It literally can't flex in the wind anymore.
Yeah, that's going to be a structural nightmare.
Oh, absolutely. As the seasons change, pressure builds, and the structure just slowly starts to fail. And, that is exactly what is happening in your myocardium, your heart muscle, after a severe ischemic event like a heart attack.
Right, because the acute crisis, the actual lack of oxygen, passes. But the heavy, stiff fibrotic tissue that gets left behind, it mechanically restricts the heart.
Right.
And that inevitably drives the patient toward heart failure. It's a progressive, compounding, mechanical failure.
And for decades, right, the clinical consensus has pretty much been that once that dense fibrotic scar forms, you're just on a one-way street.
Yeah, totally. The damage has always been seen as irreversible.
Yeah.
You manage the decline with drugs, but you definitely don't rebuild the wall.
But what if we could send in a fleet of microscopic, genetically programmed demolition robots to literally eat away that stiff cement?
That would be the holy grail.
And here is the kicker. What if, while they are chewing up the scar, they are simultaneously planting biological seeds to regrow the original flexible wood?
Sounds like science fiction.
It does. But today, we are taking a deep dive into some truly wild, paradigm-shifting science that essentially turns your immune system into that exact demolition and reconstruction crew.
I'm so excited for this one.
We're unpacking a 2026 breakthrough article from the journal Cell Stem Cell by Zhang and colleagues. And it's going to completely change how you think about cardiac regeneration.
It really is an elegant, multi-layered approach.
Yeah.
Because we're looking at an engineered cellular therapy that functions like a biological Swiss Army knife.
Oh, I love that analogy.
Yeah, it's designed to navigate directly to the heart, selectively hunt down pathogenic scar tissue, and then locally secrete factors that force mature tissue to start multiplying again.
It's so dense and brilliant. So let's get right into the pathology of the scar itself, right? Because knowing your enemy is half the battle.
Absolutely.
When a patient suffers a myocardial infarction, which is a heart attack, we know the ischemia rapidly kills off billions of cardiomyocytes, those heart muscle cells.
And because the adult human heart is notoriously terrible at regenerating, the body just hits the panic button.
It just freaks out.
Completely, to prevent a catastrophic myocardial rupture.
I feel like the heart wall literally tearing open.
Literally tearing open under the pressure of pumping blood. So to prevent that, the local environment triggers resting cardiac fibroblasts.
Okay, and fibroblasts are...
You can think of them as the dormant scaffolding crew of the tissue. They usually just quietly maintain things. But in response to all that massive cellular death and the inflammatory cytokine storm, they undergo a phenotypic switch.
They transform.
Right. They turn into a hyperactive state known as myofibroblasts.
Okay, and these myofibroblasts are the ones furiously pouring that quick, dry cement we talked about.
You got it. They secrete immense volumes of extracellular matrix, or ECM, which is heavily biased toward highly cross-linked collagen proteins.
Which forms that fibrotic scar.
Exactly. And that scar is akinetic. It doesn't contract. It doesn't conduct electrical signals. It just sits there, forcing the remaining healthy myocardium to work twice as hard.
Which causes hypertrophy and eventually failure. So the goal is obvious, right? We need to eliminate these rogue myofibroblasts.
Right.
But here's where I get stuck. The heart is full of resting fibroblasts that we actually need to maintain normal tissue structure.
Yes, you can't just wipe them all out.
Right. So how do we target just the hyperactive pathogenic ones without triggering a massive autoimmune attack on the healthy cells?
So the researchers solved this by identifying a very specific molecular bullseye. It's called fibroblast activation protein, or FAP.
FAP.
And to find this, they utilized single-nucleus RNA sequencing, or snRNA-seq.
Which sounds incredibly complicated.
Well, it's amazing technology. Instead of just looking at a mush of the whole tissue, this technique isolates individual cell nuclei. It reads the exact messenger RNA transcripts being produced in real time.
Oh, wow. So it tells you exactly what genes are turned on in a single specific cell.
Exactly.
It's like wiretapping the communication of every individual worker on the construction site to figure out who is pouring the bad cement.
That is a perfect way to look at it. And the snRNA-seq data revealed that FAP expression spikes dramatically around 7 days post-injury.
Okay.
And crucially, it is overwhelmingly restricted to those dangerous activated myofibroblasts. The healthy resting fibroblasts and the beating cardiomyocytes, they barely express it at all.
So FAP is basically a glowing neon hard hat that only the rogue construction workers wear. I love that detail.
It makes targeting so much easier.
Yeah. Okay, so it's time for a quick mini recap and bullet summary for you to lock this in before we move on.
Let's do it.
Bullet 1. A heart attack kills muscle, which causes resting fibroblasts to panic and turn into myofibroblasts. Bullet 2. They build rigid, akinetic scars to prevent the heart from bursting. And bullet 3. These dangerous cells wear a unique FAP neon hard hat.
Perfect summary.
So here's a question for you to think about. If you were a molecular engineer, knowing that this highly specific neon hard hat exists, how would you go about building a drug or a delivery system that only interacts with that exact marker?
Which is a huge challenge, and it leads us directly to the logistics of the hit.
Right.
Because identifying FAP is great, but getting a therapeutic agent deep into a heavily scarred, poorly vascularized organ is a massive pharmacological hurdle.
Yeah, and this is where I actually want to push back a bit on the current trends in the field, because everyone talks about CAR T cells, right? Oh, everywhere. It's the hottest topic in oncology.
Exactly. We engineer T cells to hunt down leukemias and lymphomas incredibly well. Why not just use DR T cells to hunt down FAP?
It's really a problem of terrain and traffic. T cells are phenomenal at surfing the blood to hunt liquid tumors, like in the blood or bone marrow.
Right.
But when they are faced with a dense, solid, highly fibrotic organ like a scarred heart, they struggle to physically penetrate that thick extracellular matrix.
So T cells are like sports cars. Great on the highway, completely useless if you need to off-road through a dense forest.
Exactly. They also suffer from exhaustion in the harsh hypoxic microenvironment of a cardiac scar. They just give up.
Okay, but what about macrophages? Because we've seen CAR-macrophages used recently to literally eat fibrotic cells.
Yeah.
And macrophages are professional phagocytes, right? They are built to consume debris.
Macrophages are powerful, yes. But they have a fatal logistical flaw when you use them as an infused therapy. They are primarily tissue-resident cells.
Meaning they don't like to travel. Right. If you engineer macrophages in a lab and inject them intravenously into a patient, they don't circulate well. They tend to get trapped in the first capillary beds they hit.
Oh, like in the lungs or the liver.
Exactly. Long before they ever reach the heart.
Man, and this is the genius pivot in the Zhang paper. They didn't use T cells and they didn't use macrophages. They used monocytes.
Yes.
Let's unpack the biology of a monocyte, because this seems to be the real linchpin of their delivery system.
Monocytes are just this elegant natural solution. Physiologically, monocytes are circulating white blood cells. They are actually the highly mobile precursors to macrophages.
Oh, okay. So they eventually become macrophages anyway.
Exactly. When the heart suffers an infarction, the necrotic tissue releases specific inflammatory signals called chemokines.
Like distress flares.
Yes. Specifically one called CCL2. And monocytes possess an innate evolutionary homing mechanism for these exact signals. They surf the bloodstream, sense that chemokine gradient, and actively migrate directly into the injured myocardium.
And then they transform.
Right. Once they arrive, they naturally differentiate into macrophages to begin the cleanup process.
They are like nimble paramedics on motorcycles. They easily navigate the traffic of the bloodstream, follow the sirens of the chemokines, and park right at the site of the emergency.
It's the perfect delivery vehicle. And the data in this paper backing up this homing ability is wild. They track these engineered cells in vivo, in live subjects.
And what did they find?
The monocytes achieved a 4.8-fold higher heart-to-liver ratio compared to infused macrophages.
Wait, almost 5 times higher?
Yes. That is a staggering improvement in biodistribution. It means the vast majority of the therapeutic cells actually reach the target organ without getting deposited in the liver.
Which drastically reduces off-target toxicity, I'd imagine.
And significantly increases the therapeutic payload right at the scar where you need it.
But wait, if monocytes are so clearly superior for reaching inflamed solid organs, why is this a novel breakthrough? Why haven't we always used monocytes instead of macrophages?
Historically, it has been a manufacturing bottleneck.
Really? How so?
To make a CAR-macrophage, you draw a patient's blood, isolate the monocytes, and then you have to bathe them in chemical factors for days to force them to mature into macrophages ex vivo outside the body.
Before you can even engineer them.
Right. But the researchers here bypassed all of that. They engineered the fresh monocytes directly, immediately after isolation.
Oh, wow. That cuts days off the timeline.
Yields a much higher number of viable cells, and it makes the therapy far more scalable for actual clinical use.
Okay, time for our next mini recap and bullet summary.
Go for it.
Bullet 1, T cells are like sports cars that can't penetrate dense scars. Bullet 2, macrophages get trapped in lung and liver traffic. Bullet 3, monocytes are nimble, blood-surfing paramedics that use chemokines to home in on the heart at nearly 5 times the rate of macrophages.
Spot on.
So here is a question to ponder. Can you think of other diseases defined by heavy, localized inflammation where a homing monocyte could be the perfect Trojan horse to deliver a therapeutic payload?
That is exactly the kind of question driving the next wave of cellular engineering. Because now that we have the perfect delivery vehicle parked at the cardiac scar, we have to talk about the weapon.
Yes. We need to turn this paramedic into an assassin.
We do.
And we do that by engineering it with a CR, right? A chimeric antigen receptor.
Correct.
This is where we really need to painstakingly break down the molecular architecture, because the source text gives us the exact genetic blueprint they inserted using lentiviral vectors. How does this weapon actually work?
Well, a CR is a synthetic receptor. It doesn't exist in nature. The lentivirus splices the genetic code for this receptor directly into the monocyte's DNA.
Okay, so it builds it from scratch.
Right. And the structure they engineered has 3 highly specific functional domains. First, at the very top, there is a CD8 alpha signal peptide.
Wait, what does that do? Is that just like structural?
No, it's actually a molecular shipping label. When the monocyte's internal machinery builds the newly coded receptor, the CD8 alpha peptide directs the cell to traffic this receptor out to the external cell membrane.
And anchor it there.
Exactly, facing outward.
Got it. So it ensures the weapon is actually mounted on the outside of the vehicle, not stuck inside.
Right. Below that is the targeting domain, the anti-FAP SCFV. That stands for single chain variable fragment.
Which is essentially just the highly specific tip of an antibody, right?
Yeah.
So this is the homing missile's guidance system that physically locks onto the FAP protein, our neon hard hat.
Correct. But here is a critical mechanical detail. Connecting that SCFV to the cell surface is a flexible hinge region.
Why a hinge?
Because cells aren't perfectly flat surfaces. The hinge gives the receptor the steric flexibility to pivot and grab onto FAP proteins that might be sitting at really awkward angles on that dense scar tissue.
Oh, that's clever.
And finally, tethered to the inside of the cell membrane, is the trigger, the CD3 zeta intracellular signaling domain.
Let's visualize this with a classic arcade game. The monocyte is Pac-Man. The SCFV on the outside is the joystick. It touches the FAP target and locks on. But locking on isn't enough. The second it binds, it sends a mechanical and chemical signal down through that hinge to the CD3 zeta domain inside the cell. And that domain is the big red button. When it gets pressed, it just screams, "Eat!"
The scientific term is phagocytosis, but yes. And the mechanics of it are beautiful.
Walk us through it.
When CD3 zeta is phosphorylated, it kicks off an intracellular signaling cascade that radically alters the monocyte's cytoskeleton. The internal actin filaments rapidly polymerize and rearrange.
Meaning the cell physically changes shape.
Radically. It causes the cell membrane to literally reach out, envelop, and pull the entire pathogenic myofibroblast inside itself to be digested by enzymes.
And this isn't just theoretical. The paper includes live cell confocal microscopy. They recorded this happening.
It's amazing to watch.
You can literally watch these engineered cells, which they call FA-Par-Mos, creep up to the FAP-positive target cells, stretch their membranes around them, and swallow them whole. It is a highly targeted microscopic demolition derby in real time.
And it is exquisitely specific. Because the CR requires the presence of FAP to trigger that cytoskeletal rearrangement, the engineered monocytes ignore the healthy cardiomyocytes and endothelial cells entirely.
They just leave them alone. Okay, it's time for our next mini recap and bullet summary.
Repair it.
Bullet 1. We engineer the monocyte with a CR that has a CD8 shipping label and a flexible hinge. Bullet 2. The SCFV joystick locks onto the FAP scar tissue. Bullet 3. The CD3 zeta trigger gets pressed, and the monocyte physically swallows the bad cells whole via phagocytosis.
Excellent.
Now, here is a terrifying question for you. We've cleared out the stiff scar tissue, but earlier we talked about how that quick-dry cement was holding the damaged house together. If we just remove the cement, what is physically stopping the house from collapsing?
That is the exact physiological paradox that ruins most antifibrotic therapies. And it brings us to the absolute genius of this paper.
Because you can't just demolish.
Right, you must reconstruct simultaneously. If you just remove the collagen matrix, you induce fatal cardiac rupture.
So they didn't just build a CAR-monocyte, they built a pCAR-Mo. And the P stands for pleiotropic.
Yes, pleiotropic means a single gene or cell producing multiple distinct physiological effects.
Multitasking, essentially.
Exactly. To achieve this, the researchers packed an extra genetic payload into the monocyte, a gene that constantly secretes a protein called Agrin.
Agrin. Okay, this is the biological seed, the miracle grow. I want to spend some time here. What exactly is Agrin, and why does it have this crazy regenerative power?
Agrin is a heavily glycosylated extracellular matrix protein. But what is really fascinating is its temporal expression.
Meaning when it shows up in life?
Yes. In mammals, it is found at high concentrations in the neonatal or newborn cardiac microenvironment.
Oh, because babies heal fast.
Essentially, yes. A newborn's heart actually retains a brief window of true regenerative capacity. But shortly after birth, the tissue composition changes, agrin levels plummet, and adult cardiomyocytes undergo terminal differentiation.
Terminal differentiation.
They exit the cell cycle forever. They simply stop dividing.
Wow. But our engineered pCAR-Mos are actively secreting this neonatal protein locally into the adult scar environment.
Exactly. When agrin binds to specific glycoprotein receptors on the surface of the surviving adult cardiomyocytes, it triggers an intracellular cascade.
What does that do?
Specifically, it involves the stabilization of the dystrophin glycoprotein complex. This biochemical signal essentially overrides the adult stop command. It forces these stubborn, mature heart cells to re-enter the cell cycle and physically divide.
And the researchers proved this wasn't just like cell swelling, right? It was true division. They tracked nuclear markers like Ki67, PH3, and ARCDIB.
Right. Those specific proteins are only synthesized when a cell is actively unzipping its DNA and undergoing mitosis. Finding them in adult cardiomyocytes is the ultimate proof of regeneration.
But hang on. If you are rapidly multiplying new muscle tissue, you drastically increase the oxygen demand in that area. If there's no blood supply, the new muscle will just die again.
Agrin solves that as well. It is highly pro-angiogenic.
It grows blood vessels.
Yes. It stimulates the local endothelial cells to sprout new blood vessels. The team proved this in vitro using HEVECs, which are human umbilical vein endothelial cells.
And what happened?
When exposed to the pCAR-Mos, these simple endothelial cells organize into complex branching networks of vascular tubes.
So the pCAR-Mo is basically a completely self-sufficient terraforming machine. It clears the dead brush, but simultaneously sprays a highly targeted fertilizer that forces new muscle to grow, while also laying down the irrigation pipes to feed that muscle.
It's a highly synchronized ballet of destruction and creation.
Oh, and I read there's a bizarre feedback loop here too, right? The agrin actually makes the CAR-monocytes better at eating.
It does. It's a brilliant synergistic effect. The secreted agrin acts on the monocytes themselves, inducing further cytoskeletal remodeling.
It changes their shape again.
It makes their internal structure more dynamic and fluid. This significantly lowers the physical energy required to engulf the dense scar tissue. The agrin supercharges the demolition while driving the reconstruction.
That is just so elegant. All right, mini recap and bullet summary time. Bullet 1. Agrin is a neonatal protein that forces adult heart cells to divide, marked by Ki67 and PH3. Bullet 2. It grows new blood vessels to feed the muscle. Bullet 3. It dynamically alters the monocyte structure to make it a supercharged scar eater.
Great summary.
So here is your question to chew on. Why is it so absolutely crucial that the monocyte delivers the agrin locally, directly into the injury site, rather than a doctor just giving a patient a systemic agrin pill to swallow?
Oh, that's a good one.
Hint, consider the consequences of turning off the stop dividing signals in cells everywhere in your body all at once.
Yeah, that need for extreme localized targeting is exactly why systemic regenerative drugs have failed in the past.
Which brings us to the ultimate proof of concept, Section 5. The theory is bulletproof. The cellular mechanics are incredibly sound. But does this highly complex engineered biological machine actually work in a living, pumping cardiovascular system?
To test this, the researchers moved to an in vivo murine model. They subjected mice to a procedure known as permanent LAD ligation.
LAD, that's the left anterior descending coronary artery, right?
Often colloquially called the widowmaker.
Yikes.
By surgically tying it off permanently, they induced a massive, highly standardized ischemic infarction in the mice.
Basically mimicking a severe human heart attack.
Exactly. They then intravenously infused the pCAR-Mos, and the clinical data they got back was just staggering.
Let's start with the macro level. The mice that received the pCAR-Mos had significantly higher overall survival rates. Furthermore, their echocardiograms showed massive improvements in LVEF, or left ventricular ejection fraction.
LVEF is the clinical gold standard for measuring heart failure. It represents the percentage of blood the left ventricle pumps out with each contraction.
So a drop means failure.
Right, a severe drop means the heart is failing as a pump. But the pCAR group showed profound functional recovery of that pumping capacity.
But the researchers didn't stop at the macro level. They dug into the microenvironment of the tissue itself. They looked up the biomechanics of the scar, specifically the ratio of collagen I to collagen III.
Because that ratio dictates tissue compliance. Collagen is incredibly thick, rigid, and stiff.
The bad cement.
Yes. While collagen III is more elastic and flexible, the pCAR-Mos therapy heavily reduced the presence of collagen I.
So it physically changed the tensile properties of the heart wall.
It transformed it from a rigid block of akinetic tissue back into a compliant, flexible structure.
But the absolute mic drop moment of this paper, for me at least, was when they applied that ultra-advanced single-nucleus RNA sequencing to the treated heart.
The send-RNA-seq, yes.
They sequenced 18,840 individual cells to see exactly what had changed at the genetic level across the entire organ. What did they find?
The data provided definitive proof of regeneration. In the pCAR-Mos-treated hearts, they observed a 4.6-fold increase in the absolute number of cardiomyocytes.
A nearly 5-fold expansion of beating muscle cells.
And they saw a downregulation of apoptosis or cell suicide pathways.
And what happened to the dangerous fibroblasts?
They were decimated. The data showed a steep depletion of the pro-fibrotic FAP-positive myofibroblasts.
For riddance.
But fascinatingly, it wasn't just a deletion. They observed a rise in a subpopulation known as MFCs, which are myofibroblast-derived cells.
What are those?
They are a deactivated healing state of fibroblasts that actually upregulate antifibrotic genes.
So the engineered monocytes didn't just clear the bad cells, they reprogrammed the entire cellular neighborhood toward healing.
Exactly.
I want to introduce a quick mnemonic to help anchor this entire highly complex process in our minds. If you want to remember how this therapy works, think of the word CARE. C-A-R-E.
I like this.
C stands for clear the scar via targeted phagocytosis. A is for Agrin delivery directly to the microenvironment. R represents regrow the muscle by overriding cell cycle arrest. And E is to enhance survival by restoring elasticity and reducing apoptosis.
The CARE mnemonic beautifully encapsulates the science.
Thank you.
But before we conclude, we really have to mention the delayed administration data, because this dictates its viability in the real world.
Oh, right. Because in emergency medicine, patients do not always receive targeted cellular therapy the moment they suffer an infarction.
It takes time. You have to stabilize the patient, assess the damage, and prep a cellular infusion. By the time you are ready, the fibrotic cascades have already begun.
So what do they do?
To mimic this clinical reality, the researchers waited 3 to 7 days after the induced heart attack to infuse the pCAR-Mos.
When the permanent scar had already established itself.
Yes, and miraculously, the treatment still exerted profound effects. It reduced the infarct size and restored ventricular function.
Proving that we are no longer just preventing damage, we possess the biological tools to actively reverse existing fibrosis.
It's groundbreaking.
All right, time for our final mini recap and bullet summary.
Let's bring it home.
Bullet 1. In mice with severe heart attacks, the therapy massively improved survival and pumping efficiency. Bullet 2. It shifted tissue mechanics by reducing rigid collagen I. Bullet 3. Single-cell sequencing definitively proved a 4.6-fold expansion in heart muscle cells. And bullet 4. It works even when administered days after the initial injury.
Perfect.
So here's your question for this section. Think about the timing. Why is the fact that delayed treatment, like day 3 or day 7, works such a game changer for emergency medicine?
Because it turns a frantic emergency into a manageable, curable condition. What this paper represents is a fundamental shift in cardiovascular medicine.
It really is.
Ischemic heart disease is a leading driver of global mortality because, until now, end-stage heart failure was a terminal decline. The only true cure was an allogeneic heart transplant.
which carries a staggering 50% mortality rate over 5 years just waiting on the transplant list.
Exactly. But by taking a patient's own circulating monocytes, engineering them with a highly specific FAP targeting CRR, and weaponizing them with regenerative Agrin, we are mounting a preemptive strike.
We are taking that one-way street of heart failure and paving a road backward toward healthy tissue.
It's remarkable.
It is. But I want to leave you with a provocative thought that pushes beyond cardiology. Because the authors of this paper explicitly note that this dual-function platform, the ability to selectively eat a scar and simultaneously secrete a regenerative factor, is a plug-and-play system.
Right.
And fibrosis is the final, fatal pathway for dozens of incurable diseases.
The implications are truly boundless. The biological architecture of a scar in the heart shares incredible similarities with scars in other organs.
Exactly. So what happens if we take this same engineered monocyte, but we program the delivery payload for the lungs to treat idiopathic pulmonary fibrosis?
Or if we target the kidneys.
For a liver that has been completely destroyed by cirrhosis? Could we be looking at a future where a simple IV infusion of your own modified cells could quietly patrol your bloodstream, hunting down and eating away the internal scars of aging and disease across your entire body?
It's a profound thought.
It makes you wonder, if you control this technology right now, what organ system would you target next? Think about it, dive into the research, let this spark your curiosity about the incredible future of cellular engineering. And until next time, keep digging deeper.